Hello everyone, and welcome to the new webinar series. My name is Tracy Ying. I work on the marketing team here at GenScript, and I'll be your host for the upcoming 7 webinars. They are very excited to have guest speakers from NIH, UMass Medical School, UCSF, and the University of Minnesota. We will cover interesting topics in CRISPR, bispecific antibody inject discovery, T-cell engineering, HIV vaccine development, and more. And here on this slide, you can see a complete list of our 7 webinars, topics, and speakers. Before I hand over to today's moderator, Dr. Claire Zhou, I have a few housekeeping items to cover about this presentation. First, today's webinar will be available on demand after the live session and is accessible on GenScript website. We will upload the slide deck as well. Next, we'd love to hear from you during today's presentation. If you have a question for our speaker, please feel free to send it through the chat box at the bottom of your player or by tweeting us at GenScript. We'll be answering questions at the end of the session. If we don't get to your question during today's webinar, we will be sure to follow up afterward. And last, we'd like to encourage you to complete a quick survey after this webinar and share today's webinar with your social networks. If you mention your colleagues that you think will benefit from this webinar, both of you will get 1,000 easy coupon points from us as a thank you token. So without further ado, I'd like to kick off things by welcoming our moderator, Dr. Claire Zhou. Over to you.
Hi, everyone. This is Claire Zhou. Welcome to the first webinar of GenScript's 2018 Fall Webinar Series. I'm the marketing specialist at GenScript, and it's my pleasure today to introduce Dr. Shang Zhou as our speaker. Dr. Shang Zhou obtained his PhD degree in molecular biology and genetics from the Chinese Academy of Science in 2012, focused on studying the mechanism of how steroid hormone regulates IIS TORC1 signaling in insect fat bodies. Using molecular genetic tools and techniques, including DNA cloning, gene overexpression, and RNAi, and Drosophila genetic manipulation, Dr. Zhou did 2 years of postdoc training in biomedical engineering at Tufts University, using molecular method to engineer recombinant spider silk to facilitate osteogenesis in vitro. Since 2014, Dr. Zhou works as a senior scientist at GenScript, and he has established a CRISPR-based genome editing cell line engineering platform, which he has delivered more than 500 CRISPR cell lines. Dr. Zhou has plenty of hands-on experience on CRISPR design, and today he will share with us some practical tips for effectively generate CRISPR knockout or knock-in cell lines. If you have any questions during the presentation, you can submit them by typing them in the question box that you can see on your screen. We will have a Q&A session at the end of the talk. So with this, I'll pass the presentation over to Dr. Shang Zhou.
Hi, I am Shang Zhou. Today I'm going to present a topic, how to effectively use CRISPR in cell line engineering. In this topic, I will focus on optimized workflow and tips. My presentation are divided into 3 parts. At first, I will briefly go over CRISPR background. CRISPR-Cas9 is an RNA protein complex composed of guide RNA and Cas9 protein. The RNA harboring the sequence that is complementary to DNA sequence, so the RNA can guide the Cas9 protein to a specific site on genome. The gRNA Cas9 complex binds the specific DNA and cut the double-stranded DNA. Most importantly, the Cas9 only binds the DNA sequence to PAM motif, that is the NGG. The cleavage site is always 3 to 4 base pair up to PAM. What if we deliver the gRNA Cas9 complex into live cells? We can deliver the gRNA Cas9 into live cells through plasmid-based transfection, virus-based infection, or ribonuclear protein-based microinjection. The gRNA Cas9 form complex and enter cell nucleus. Then the complex cut the genome at a specific site. The DNA double-strand break induces DNA repair response in the cells and can be repaired in two different pathways. Mostly, it is repaired through NHEJ pathway that generates insertion or deletion mutation at the specific site. If the specified site locates at the protein coding area, the protein function can be knocked out for frame shifting mutation. It is also can be repaired through HDR pathway when a donor DNA is given. In this case, DNA modification and insertion occur at the specified site called the knock-in. The CRISPR-Cas9 technology are mostly used at 4 application fields. For drug discovery, people use this technology to identify and validate new drug targets, as well as to engineer cell or animal models. For industrial bioproduction fields, people use this technology to develop bioprocess cell line or bacterial strains for drug or biomaterials production. At agriculture field, people use this technology for plant breeding. At present, the technology are more and more attractive for gene therapy and cell therapy. For example, people use CRISPR to engineer CAR T cells. In the CAR T cell therapy, patients' PBMC are collected and the T cells are isolated. Then the T cells are engineered to express antigen-specific TCR or CAR. The engineered T cells are expanded and injected back to patient's body. Then the engineered T cells kill the cancer cells. The CAR T cell therapy presents good effects to cure blood cancer. With CRISPR technology, people can make the CAR T cell therapy cheap and robust. For example, development of off-the-shelf CAR T cells by knockout of TCR or B2M gene. Knockout antigen gene in T cells to prevent self-killing effects, and enhancing anti-tumor activity of CAR T cells by knockout of immune checkpoint inhibitors, for example, PD-1 or LAG-3. Until now, there are 11 clinical trials of cell therapy are reported to use CRISPR technology. And more recently, one paper reported to engineer non-viral TCR or CAR T cells with CRISPR. The author successfully replaced the endogenous TCR with the antigen-specific TCR by transfection of RNP and DNA donor. GenScript invites the author to present a webinar about this fantastic technology on November 1st. So please do not miss it if you are very interested in this topic. Next, I will talk about an optimized workflow for CRISPR cell line engineering. The CRISPR knockout or knock-in workflows can be separated in 5 phases. Host cell line characterization is the first phase and is important for success of the project. Within this phase, within the first phase, we must do mycoplasma test, transfection, and clonality optimization. Phase 2 is gRNA Cas9 design and synthesis. The phase 2 can be done concurrently with phase 1 as long as the promoter validation is determined Within this phase, selecting an appropriate gRNA delivery system is critical for success of the projects. The phase 3 is to transfect the gRNA, Cas9, and a donor into cells to generate knockout and knock-in cell pools. Within this phase, we do enrichment of transfected cells in order to enhance the knockout or knock-in efficiency. The phase 3 is a checkpoint in the cell pool. If more than 20% of gRNA cleavage efficiency is detected in the cell pool, we will go ahead. If less than 20%, we will stop and troubleshoot what happened. So this phase is very important for the whole project. So it's like a checkpoint. The phase 4 is to generate single clones. So after the checkpoint, if everything looks good, so go ahead to generate the single clones. You can use limited dilution or factor sorting. The phase 5 is to identify knockout and knock-in clones. We mostly use Sanger sequencing to screen the clones. It is fast and high throughput. So after Sanger sequencing, after we identified some knockout and knock-in candidates, then we will use RT-PCR, qPCR, Western blot, or functional assay to do further validation. So the host cell line characterization is the first phase and is important for success of the project. So number 1 is the mycoplasma test. So the host cell line for CRISPR editing must be mycoplasma free. You can test the mycoplasma using the PCR-based method or using the whole transfer DNA staining. Number 2 is for cell culturing. Please select an appropriate host cell line for your projects and adjust the cells to good condition before transfection of gRNA and Cas9. So if you have alternative cell lines that you can select, please select a easy to handle cell line. That will make your project more smoothly and easier. Number 3 is optimization of transfection. So test the host cells with different transfection reagent and methods. So you can choose the best one. And then select an appropriate delivery method. So that is the plasmid or lentivirus or RNP. For plasmid or lentivirus-based methods, the gRNA and Cas9 expression are driven with a promoter. So the expression should be driven with a strong promoter in your host cells. So before initiating the project, you'd better validate your promoter. Clonality optimization. For some cell lines, the clonal efficiency is low. So in this case, you'd better test and select an appropriate method. So you can try limited dilution or factor sorting for single cloning. Alternatively, you can use conditioned medium or single cloning enhancing reagent. You can purchase the enhancing reagent from vendor, for example, the stem cell. And for the cancer cell lines, the cancer cell line often carry gene copy number variation, and often the gene may not be diploid. So in this case, you'd better to investigate the gene copy number before your experiment. So you can investigate the gene copy number via a database, for example, the COSMIC, Or you can also investigate by experiment using the ddPCR. The gRNA Cas9 can be delivered to cells mainly in 3 different forms: plasmid, lentivirus, or RNP. RNP is the synthetic gRNA plus the Cas9 protein. So the plasmid-based method is very popular and cheap, but it is only best for knockout and knock-in in easy to transfect cancer cells. Lentivirus and RNP show advantages in hard to transfect cells, but lentivirus integrates Cas9 on genome and has a high off-target risk. RNP showed an extensive advantages for knockout and knock-in in any types of cell lines, especially for primary and stem cells. So you can choose the appropriate delivery system according to this table. So if you choose a good delivery system, that means your project will be much easier. The cell pool stage is a checkpoint through the whole project. So you'd better choose an appropriate method to detect the cell pool. So the TIDR is a method using web-based software to deconvolute single sequence data. The method is most popular and can be used for detection of indel or point mutation efficiency. So the software is free to use. The T7E1 and RFLP both use in the nucleus to identify the knockout or knock-in. The method is fast but with low sensitivity. Applicant sequencing and qPCR are with high sensitivity, but the cost is high. For knock-in experiments, they are very useful to determine the accurate HTI efficiency. FACS is also very useful to detect knock-in insertion of fluorescent marker genes, for example, GFP, or target protein with a specific antibody. So choosing an appropriate method to examine the cell pool for knockout and knock-in efficiency will determine the success of your projects. In order to promote HDI efficiency, in this case, we used the RNP and asymmetric single-stranded DNA donor. We made a silent mutation on homology arm to introduce Hint3 recognition site for cell pool examination and clone screening. We transfected the cells with the electroporation. Asymmetric single-stranded DNA is reported it can increase HDI efficiency. To design an asymmetric single-stranded DNA donor, the Cas9 cleavage site with a 36 base pair on the palm distal side and with a 91 base pair extension on the palm proximal side of the break. And the single-stranded DNA is complementary to the gRNA non-target strand. And in the single-stranded DNA donor, so 2 nucleotides at either 5' or 3' termini are modified with the PTO. So for more details about this asymmetric single-stranded DNA donor, you can send an email to us or refer to the published paper. Here is the third case study we knock in GFP insertion in HEC293 T cells. In this case, we use the long double-stranded DNA and a long single-stranded DNA donor. Donor and the RNP were transfected to cells with lipo 2000, and then we examined the GFP expression by FACS in the cell pool. We obtained 45% of GFP positive clones with double-stranded DNA and 32% with a single-stranded DNA donor. For knock-in insertion experiments, double-stranded DNA and a single-stranded DNA donor show different advantages. Double-stranded DNA confer higher HDR efficiency, but with high off-target efficiency. Single-stranded DNA is with a lower off-target effect and lower toxicity. So, for easy cancer cell lines, for example, HEK293 T cells, double-stranded DNA donor is good enough. For suspension, primary, and stem cells, single-stranded DNA can be good choice for its low toxicity. So GenScript provides services for both double-stranded DNA and long single-stranded DNA. If you are interested, please send an email to GenScript. Here I will draw a summary about how to effectively generate CRISPR knockout and knock-in cell lines. So choose the best gRNA-Cas9 delivery method for your host cell line. So using a best transfection method. So choose from the lipo electroporation or lentivirus. So using a best gRNA-Cas9 form, you can use plasmid, lentivirus, or RNP. So choose the best gRNAs for your target gene. It is the best to using validated gRNAs. You can find the validated gRNAs in published paper, or you can test the gRNAs before your experiments. So enrich the transfected cells. You can use the factor sorting or antibiotic selection. In order to increase the HDR efficiency in your host cells, you can use the asymmetric single-stranded DNA for point mutation and using long double-stranded DNA donor for gene insertion. Next, I will talk about some issues we have had among CRISPR experiments. The issue 1. Low gRNA cleavage efficiency is detected in transfected cells. So the issue may result from the below reasons. Reason 1 is low transfection efficiency. For this reason, you'd better optimize the transfection efficiency with a positive control gRNA or Cas9 GFP plasmid or lentivirus RNP, and enrich the transfected cells by FACS sorter or antibiotic selection. The reason 2 is low gRNA cleavage activity. So you'd better design multiple gRNAs and target different exons, so you have options to choose the best gRNAs. Check the gRNA sequence whether the PAM is contained in plasmid and synthetic oligos. So this case sometimes will happen to new people. So you'd better to not you cannot contain the PAM in your gRNA sequence in the plasmid or synthetic oligos. So in order to increase the gRNA cleavage activity, you can use single-stranded RNAs for RNP. You also need to use the N-terminal and C-terminal tagged NS, Cas9 plasmid or protein. So the reason 3 is the cell death or growth inhibition after gene knockout. So it is important to investigate phenotypes of gene knockout or knockdown through papers before experiments. If cell death or severe growth inhibition, inducible knockout or knockdown is recommended. If knockout slow cell growth rate, you can detect gRNA cleavage efficiency at 24 hours to 96 hours after transfection, and then detect the gRNA cleavage efficiency, selects the highest time point to plate the single clones. So the issue two is low HDR efficiency is detected in transfected cells. So the issue may result from the below reasons: the low gRNA cleavage efficiency. Before the HDR occurrence, the gRNA-Cas9 must cut the DNA. So if the gRNA cleavage efficiency is low, HDR must be low. So for this reason, refer to the troubleshooting of low gRNA cleavage on the last slide. So the reason two is the sequence difference between the genome and the homolog arms at the donor. So for this reason, because when we design the donor, we always download the sequence from the NCBI database. But sometimes the real sequence may be different from the NCBI database. So it is better to sequence the homolog arms from the genome before synthesize the donor. Reason three is HDR pathway activity is low in your host cells. So it is best to using an alternative host cell line that has been used for HDR in published paper. So you can increase the HDR activity by using some drugs like NU7441, NOC, and SCR7. These drugs increase the HDR by inhibits NHEJ pathway or collect the cells at the same stage with high HDR activity. So test the drugs effect before the selection, because some of the drugs are toxic to your host cells. Introduce a silent mutation on donor to avoid the gRNA recutting. Using the gRNAs that are very close to the mutation sites, so less than 15 base pair is recommended. For insertion, using PCR amplified or restriction endonuclease linearize the double-stranded DNA with long homolog arms. For point mutation, using asymmetric single-stranded DNA donor. Issue 3. No expected function is observed in knockout or knock-in cells. So the issue may result from the below reasons. So the reason one, the truncated protein still works to maintain the function of the original protein. So using gRNAs that have been validated to disrupt function of the protein in published paper may solve this problem. And design gRNAs to target the area that is homolog to the transgenic mouse model. Because some database, for example, the Jackson Lab database, provides a lot of transgenic mouse model that has knockout the genes. So there are a lot of experimental data to indicate which area is important. So design the gRNAs to target the upstream of the functional domain. So the reason two is that alternative spliced isoforms works to supplement the function of the original protein. So for this reason, using double gRNA strategy to delete the gene from the genome. Reason three is homolog or multiple copy genes supplements the function of the protein. So before your experiment, you would better investigate the homologous or multiple copy of genes in host cells by informatics analysis or whole genome sequencing, or using some database, for example, Cosmic. And design the gRNAs to target homologous genes. Reason 4 is the endogenous gene expression level is very low. So this reason is used to account for some fluorescent, fusing the fluorescent genes like GFP at the N-terminal or C-terminal of the endogenous genes. So if you cannot detect the GFP expression, that means endogenous gene expression is low. So in this case, you'd better investigate the gene expression level before selection of the host cell line. And also you can use some drugs to stimulate the expression of the gene. So at GenScript, we provide fully validated gene knockout or knock-in cell lines and cell pools using CRISPR technology to help make your research easy. Our GenScript cell line service platform is a one-stop solution from design to functional access. We have delivered hundreds of stable cell lines, and we can deliver as fast as 11 weeks. With our versatile gRNA-Cas9 vector and RNP system, expertise with various gene transfection methods, as well as PhD-level customer support, GenScript is dedicated to help make genome editing easy for you. Besides our cell line generation service, we also provide basic CRISPR reagents, including either gRNA-Cas9 plasmids or the RNP system. Our gRNA-Cas9 plasmid is validated by the Broad Institute of MIT and Harvard. We provide either both the dual vector and the all-in-one vector system for sgRNA expression. So in addition, we also have SAM constructs for transcription activation. For the RNP system, we provide fully customizable synthetic sgRNAs, unmodified or modified. We also have synthetic custom cRNAs and trace RNAs, as well as synthetic single-stranded DNAs up to 8,000 nucleotides long in length for serving as HDR DNA donor templates. In addition, we also provide the SP-Cas9 nuclease with 2 NRS for optimized nuclear compartmentalization, and the Cas9 protein with 1 NRS and an EGFP marker enabling fluorescent activated cell sorting. For more information, please visit our website listed here. Our website also provides various technical resources that can help make your research easy. For example, you can check out our new CRISPR RNP user manual or our comprehensive guide on CRISPR handbook for free. We hope you find these resources helpful. Thank you.
Thank you, Sean, for your wonderful talk. For more information, please visit our website at www.genscript.com, or you can directly email me at clairez@genscript.com. I would also like to mention that we have another CRISPR webinar coming up on November 1st, which will be delivered by Theo Rose from UCSF. The topic for this upcoming webinar will be focusing on their lab's recent studies on how to use CRISPR for large knock-ins in human T cells using non-viral HDR templates. We're looking forward to seeing you there as well. Now we'll start our Q&A session. Please type in your questions in the question box if you haven't done so already. Finally, at the end of the presentation, you will receive a brief survey. We encourage you to fill out the survey, as this will help us better design and optimize our service and webinar to meet your needs in the future. With that, we already have received some questions. Let's start our Q&A session. So the first question is, when I engineer CAR T cells, what's the advantage of RNP over lentivirus? Hi, Dr. Shangzhou, can you hear us?
Yes. So, can you please repeat your question again?
Sure. The question is, when I engineer CAR T cells, what's the advantage of using RNP over lentivirus?
Okay. So for CAR T therapeutics, safety is a big concern for the patients. So the lentivirus delivers gRNA and Cas9 efficiently, but generates random insertion of gRNA Cas9 on genome. In addition, insertion of gRNA Cas9 will make gRNA Cas9 constitute expressing in T cells and confer a high risk of off-target effects. With the RNP, there are no random insertion and no constitutive expression, so RNP is more safer than lentivirus.
Thank you. Another question. When I try to design gRNAs for my knock-in point mutation experiment, I cannot find a good gRNA with high specificity. How can I knock in the point mutation without off-target?
So in this case, I would suggest to use Cas9 nickase for your experiment instead of wild-type Cas9. The Cas9 nickase does not make DNA double-strand break, so has very low off-target effects. However, Cas9 nickase has a little lower knock-in efficiency than wild-type Cas9. So when you use Cas9 nickase, you'd better to make sure the gRNA and the transfection efficiency are the best.
Thank you. Another question. What's the best size of the homology arm on DNA donor template for knock-in insertion experiments?
The homology length is dependent on the size of the insertion fragment and will need to be tested. We tested the homology arm length from 300 base pair to 800 base pair for knock-in insertion of around 1 kilobase pair fragments, and all of them can work without showing much difference. But for better efficiency, a long homology arm with more than 500 base pair is suggested on each side.
Thank you. And then we have a couple more questions. The question is, if you knock in GFP at the N terminal to the gene of interest, can we select the gRNA 80 BP away from the start codon of the gene or before the start codon of the gene? How many gRNAs are needed for the insertion of the GFP at the N-terminus region?
So for this question, actually I would suggest to choose the gRNA that is very close to the ATG. So I think if you do not have a choice, the gRNA at 80 base pair away is available, but the question is that you may get lower knock-in efficiency.
Thank you. Another question, what's your electroporation condition?
So generally, we will test the multiple voltages. For example, we use the instrument called Selectrix. So this company provides an instrument that is very easy to use. So what we need to do is just choose different voltages. So for this company has some data collection. So for different types of cell lines, so we can get some reference voltages. So it is just follow the menu. It is fine.
Sure. Another question, do you have any recommendations for minimizing off-target effects of Cas9 and the best methods to test or detect off-target effects?
Sorry, can you repeat the question again?
Yes, sorry. So the question is, do you have any recommendations for minimizing off-target effects of Cas9 and the best methods to test or detect off-target effects?
Okay. So in order to avoid the off-targets, so the best way is using the nickase Cas9. So using the nickase Cas9, you use the design the two gRNAs. So you can use the MIT's the gRNA design tool. So design the nickase gRNAs for nickase. When you use the wild type Cas9, it is very hard to minimize the off-target effect unless you can use the RNP. So for the Cas9 wild type, you use the RNP can minimize the off-target effects in your cells, but it is still cannot say that the off-target effects will be avoided. So if you have to use the wild type Cas9, I would suggest you use the RNP. And for the detection of the off-target effects, so mostly we detect the off-target sites that is predicted by the design tool, and then sequence the targets, and then check if the off-target modified. But if you want to detect the off-target effects very much, so you will need to use the NGS to do a whole genome sequencing.
Thank you. We have another question related to that. How about using a high fidelity Cas9 to avoid off-target effects? Is a nickase better?
Yeah, nickase will be better.
Sure. We have many more questions coming from our audience. And if we didn't get a chance to answer your questions, we will definitely follow up with you with emails to make sure that you get the answers that you will be needed. We see a lot of them are for methods or protocols that we use. We do have resources online that can help you with that. So we will be sending them out to you through emails as well. And let's just take one last question. So the question is, when we clone cell lines, we often see different looking clones. Would it be possible to get a knockout cell line in uncloned populations by inserting a lentivirus and selecting for transducted cells? What's the % of cells would be expected to have a gene knocked out? So to repeat the question is, when we clone cell lines, we often see different looking clones. Is it possible to get a knockout cell line in uncloned populations by inserting a lentivirus and selecting for transducted cells?
So without a cloning of the population, so you will only get a cell pool. So I do not recommend do like this to get so for preliminary tests you can do like this. So it's like that in the cell pool, maybe you can get more than 90% of clones with knockout, but you can still have less than 10% of cells may be wild type or not knockout, because the gRNA generates the indel mutations. Sometimes you will get the 3 base pair insertion or deletion. Actually, this indel is not real knockout. So I think you will not get 100% of the knockout.
Thank you.
But when you're using the lentivirus, using some antibiotic selection, you will get a high knockout. Generally, we get more than 80%.
Thank you, Dr. Zhao, for your answers. We have many more questions that we just have to follow up through emails. Thank you all for your questions and tuning in for our webinar. And now I'll hand it back to Tracy.
Thanks, Claire and Sean, and thank you all for attending. We hope you find this webinar useful for your research. Again, we would love to hear your feedbacks, so please help complete the survey after the webinar, and we appreciate your sharing this with your social networks. Thank you, and have a nice day. Bye-bye.
Bye.